This protocol describes a reliable and efficient reverse genetic system for making recombinant rotaviruses. It also explains how to make recombinant rotaviruses that express fluorescent marker proteins.
This method is simple, requiring a minimum number of plasmids, and can generate recombinant rotaviruses that express separate foreign proteins as well as all the viral proteins.
Begin by seeding BHK T7 cells onto 12-well plates. Rinse a freshly confluent monolayer of cells with PBS. Then, disrupt the monolayer with trypsin EDTA solution. And resuspend the cells in 5 milliliters of GMEM complete medium. Determine the concentration of viable BHK T7 cells using trypan blue and an automated cell counter. Then seed 2 × 10⁵ cells in 1 milliliter of GMEM into each well of a 12-well cell culture plate. Incubate the plate at 37 degrees Celsius in a 5% carbon dioxide incubator overnight. On the next day, prepare the plasmid mixture for transfection according to manuscript directions. Then add 110 microliters of pre-warmed reduced serum medium to each plasmid mixture and gently pipette up and down to mix. Add 32 microliters of transfection reagent to the mixture. Do a brief centrifugation following vortexing and incubate at room temperature for 20 minutes. Meanwhile, rinse the BHK T7 cells with 2 milliliters of GMEM in complete medium. Add 1 milliliter